Hi, my name is Sierra Caldwell, and today I'll be talking to you about the article Prion propagation and toxicity in vivo occurs in two distinct mechanistic phases. Creutzfeldt-Jakob disease is one of the few human transmissible spongiform diseases in the world, which is caused by the presence of a abnormal isoform of a cellular glycoprotein in the brain. These cellular glycoproteins are commonly referred to as proteinaceous infectious particles, or prions, which are mutated, highly infectious forms of a normal brain protein, PRPC, which are composed exclusively of PRP scrapies, or PRPSC. The aim of this experiment, which was conducted by Marlon Sandberg and colleagues, was to answer the question, does prion propagation and toxicity in vivo occur in two distinct mechanistic phases after PRPC is mutated into an infectious form? In order to answer this, they infected three groups of mice intracerebrally with RML mouse prions and studied the prion titers that occurred over time, where the measurement of prion protein concentration was achieved by the application of cell-based assay. The first group of mice was a normal wild-type strain, while the second group expressed PRP at a level 8-fold higher than that of wild-type mice. Lastly, the third group of mice were homozygous for the PRP gene, had no wild-type allelic counterpart, and expressed only half the normal amount of PRP. These mice were then killed at defined points at the onset of clinical disease. The brains were then removed and divided sagittarily, half frozen to -70°C and half fixed in 10% formal buffered saline. Prion titers were then calculated using the Reed-Mensch formula and results plotted graphically. The results obtained demonstrated that prion infectivity indeed does occur in two distinct mechanistic phases. All three groups of mice, irrespective of initial concentration of cellular PRP, increased exponentially in the clinically silent incubation stage until prion protein concentration reached its maximum. The second group of mice, TG20, reached its plateau first, shortly followed by the normal wild-type strain group and homozygous group. Phase 1 increased exponentially, however, it distinctly reached a maximal prion titer upon which it switched to a plateau phase. Figure 1.0 represents this. In Figure 1.1, clinical onset only occurred in each group after the toxic threshold was crossed, with the second group, TG20, reaching this threshold first, again shortly followed by the normal wild-type strain and homozygous group. Thus, due to these results, it can be concluded that prion propagation and toxicity in vivo indeed does occur in two distinct mechanistic phases, the clinically silent incubation phase and the extremely aggressive clinical phase. This discovery of prion infectivity and toxicity occurring in two phases has led to further research by other scientists. In 2013, Hanoi and colleagues further discovered that various prion strains all resulted in the two-phase prion infectivity and toxicity model. However, unlike the experiment conducted by Sandberg and associates, Hanoi and colleagues used three groups of the same expression mice which were infected with a variable or different prion strands. Thank you for listening to my speech.